Hello everyone, welcome to the ITLS Academy. Today the topic that we are going to discuss is the determination of the Salmonella. We all know that Salmonella is what? It is a microorganism. Okay, and this microorganism when it is contaminate the product, then it will harm the health of the it will harm the health of the human being. So it will harm the health of the human being. That's why it is very important to detect the salmonella present into a water. Okay, so in today's lecture we are going to discuss about the detection of the salmonella by using a culture media. So let's start today's lecture. Introduction. We all know that the salmonella is a gram negative bacteria. That means that when we perform a gram staining, then it will give a pink colony. That's why it is called a gram negative bacteria. It is also a oxidase negative non-spore forming rod shaped bacteria which is able to ferment the glucose and capable of facilitating the anaerobic growth. This is the introduction of today's lecture. Now come to the next slide, that is your principle. We all know that detection of the salmonella is totally based on a pre-enrichment, selective enrichment, which are followed by the isolation on a selected media. Presumptive salmonella colonies are then confirmed by the different tests, that is called a biochemical test and a serological test. Okay, first we perform a pre-enrichment test, then we perform a selective enrichment test, which is followed by the isolation on a selective media. After that, the salmonella colonies are confirmed by the two different tests, that is called a biochemical and serological test. Enrichment broth is necessary to enable injure the cell to grow. Further selective enrichment is done so that it will increase the proportion of the salmonella in relation to the background flora. Selective media are then used for a further isolation and a preliminary confirmation. Okay, now the next is your now culture media. Culture media means that a substance which is used for the growth of the microorganism. Okay, so here the culture media that we are going to use is a buffered peptone water, Rappaport Bacillades broth, that is also called a RVS broth, brilliant green phenol red lactose agar BGA, xylose lysine deoxycholate agar XLD, and bismuth sulfide agar. This is your optional and nutrient agar. Now see the composition of different culture media. Okay, so the first composition that we are going to see is a BPLS, that is your brilliant green phenol red lactose sucrose agar media. It contains peptone. Okay, so the composition that I'm going to tell you is for a 1 liter. Okay, so amount of the peptone is 5 gram. Meat extract 5 gram. Sodium chloride 3 gram. Disodium hydrogen phosphate hydrogen phosphate, it is 2 gram. Lactose 10 gram. Phenol is 0.08 gram. Sucrose is around 10 gram. Brilliant green is around 0.0125 gram, and agar The content of the agar is around 12 gram. Okay, now the next composition is for a RV broth. RV broth. Okay, so this RV broth contain a soy protein, soy peptone 4.5 gram. Okay, this composition is again for a 1 liter. Soy peptone 4.5 gram, sodium chloride, it is around 7.2 gram, potassium phosphate. Potassium phosphate. Okay, potassium phosphate is around 0.6 gram. Okay, dipotassium phosphate, dipotassium phosphate, it is around 0.18 gram. Magnesium chloride, it is around 13.58 gram. Then malachite green, it is around 0.036 gram. Okay, now the next composition is for the xylose xylose lysine deoxycholate, that is also called a XLD. Okay, again this composition is for a 1 liter. It contain agar 13.5 gram, ammonium ferric citrate, ammonium ferric citrate, it is around 0.8 gram. Next is your lactose 7.5 gram, L-lysine hydrochloride, it is around 5 gram, phenol red 0.8 gram, 0.08 gram, sodium chloride 5 gram, sodium deoxycholate, sodium deoxycholate, it is around 2.5 gram, sodium thiosulfate, okay, amount of sodium thiosulfate is around 6.8 gram, sucrose 7.5 gram, xylose 3.5 gram, yeast extract 3 gram. Okay, now the next composition is your nutrient agar, that contain nutrient agar that contain a like your beef extract, peptone, beef extract, peptone, sodium chloride and agar, and the composition we are already discussed about it. After that, what you have to do, all these media prepare separately, and after preparation of each media, what you have to do, you have to autoclave it. During autoclavation, what you have to do, you have to set the temperature 121 degrees Celsius, pressure will be 15 psi, and time will be at 20 minutes. Okay, then the next is your first is your pre-enrichment. Okay, so in pre-enrichment, you have to clean the surface of the bottle, pouch, cup with the help of the 70% of the ethanol, and the pouch or the surface of the bottle at which the sample are present. Now you have to filter the 250 ml of the sample with the help of the membrane filter, whose pore size will be a 0.45 micrometer, by using a sterile membrane filtration assembly. Now you have to place that filter disc into a 50 ml of buffer peptone water and incubate at 36 degrees Celsius for 16 to 20 hours. Okay. One more thing, you have to clean the laminar, and you have to autoclave the glassware. Okay, now what you have to do, first of all, you have to clean the bottle at which the sample are present with the help of the 70% of the ethanol. Now you have to filter that sample with the help of the sterile membrane filtration assembly, and the pore size of that membrane filter will be a 0.45 micrometer. Then you put that filter disc inside a 50 ml of buffer Peptone water and incubate at 36 plus minus 2 degrees Celsius, and the time for the incubation is around 6 to 20 hours. After that, the second procedure is your selective enrichment, in which you um, take a 0.1 ml from the pre-enrichment culture and inoculate it into a 10 ml or 1 ml to 100 ml of malachite green or magnesium chloride. This is a modified form of the RVS broth, and then again you have to incubate it for a 18 to 24 hours at 48 plus minus 0.5 degree Celsius. Now the next is your confirmation on a selective agar media. After incubation, streak a loop full from RVS broth on a selective media, that is a BGA, XLD or BCA. Okay, now what you have to do, suppose that this is your RVC broth in which there is a colonies are there. Now this is your BGA broth, this is your XLD broth. Now you have to take a one colonies from the RVS broth and make a streak on a BGA and a XLD broth. Then after that you have to incubate that plate for a 24 hours at 36 degrees Celsius. If you are using a BSA, then in that case you have to incubate it for a 48 hours. Then in order to detect the slow growing Salmonella, inoculate BGA, XLD and BSA optional after continuous incubation of RVS broth for further 24 hours. Observation: colonies on a BGA or are red or slightly pink, white and opaque with red surrounding. Okay, suppose that this plate is for a BGA agar. Now in that case you have found that a red or a slightly pink, white, even a opaque with red surrounding colonies, and the colonies which we get on XLD agar are colorless, but they are appear red, usually with a black center. And the colonies when we are using a BSA agar, then in that case we get a colony is black in color and it is surrounded by a metallic sheet. Salmonella H2S negative strain and appear on XLD agar as pink with a darkest center. Lactose positive Salmonella strain grow on a XLD agar as a yellow with or without blackening. Now the next is your confirmation test. If there are a typical suspended colonies are there, then you have to plate at least one selected colonies from each positive agar media and further four colonies. If the first colony test negative on nutrient agar, plate and then incubate it at 36 degree Celsius for 18 to 24 hours. Thereafter proceed for a further biochemical and serological confirmation. Biochemical confirmation: the first is your lactose glucose fermentation and hydrogen sulfide formation. Now in that case you have to streak a colony on iron 2 sugar agar and stab the bud in and then incubate it for a 24 hour at 36 degrees Celsius. If salmonella colonies are there, then it will show a red slant with a gas formulation and yellow bud with the blackening of the agar. Next is your urea degradation. In that case you have to incubate that colony on a slant of urea agar and then incubate it for a 24 hour at 36 degree Celsius. If there is a salmonella colonies are there, then it will show a negative reaction, that is no rose pink color followed by a diphtheris. Next is your lysine decarboxylase medium. In that case you have to inculate the colonies just below the surface of the liquid lysine decarboxylase medium and overlay the medium with a sterile liquid paraffin or oil and then incubated at 36 degrees Celsius for 24 hours. If Salmonella collies are there, then it will show up purple in color. So this is a diagram which show a biochemical characterization of the Salmonella: lactose, glucose, hydrogen sulfide, urea, and lysine decarboxylase reaction. Okay, so this Salmonella give a negative lactose test, positive glucose test, positive hydrogen sulfide test, negative urea test, and positive lysine decarboxylase test. Serological test carry out slight agglutination so that to check the presence or absence of the Salmonella. O negative VI and H antigen with the appropriate antisera from a pure colonies on nutrient agar as per manufacturer instruction after the elimination of the auto agglutinable strain. Okay, then after that expression of the result, it is simply test for a Salmonella is equal to present or absent per xml of the sample. So this is all about the detection of the Salmonella by using a culture media. I hope you will understand. But if you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. Or you can also ask your query to the helpline number that are already mentioned in your site, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.